Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And so far in the previous lecture, we have discussed about the basic principle of ion exchange chromatography, and in addition, we have also discussed about the how to perform the generalized ion exchange chromatography, what are the different steps you have to take to successfully complete the chromatography, and how to choose the different types of matrix and what is the importance of isoelectric point, and as well as the other parameters which you have to consider while you select the suitable matrix for protein purifications. In addition, we have also briefly discussed about the several applications of the ion exchange chromatography for the industrial usage as well as for the daily uses. So, in that context, we have also discussed about the protein water purifications, how the ion exchange chromatography can be used to purify the different components of the water, so that the water will be free of the salt and the heavy metals and fluorides and all the kinds of contaminants. So, now in today's lecture, we are going to discuss about the how ion exchange chromatography can be utilized to solve the different types of research problems. So, one of the major application or the utilization of ion exchange chromatography is to purify the or to process the complex protein mixture, and from there you can be able to select the protein of your interest simply using the ion exchange chromatography. So, we will go through with the several examples how you can process the samples and what are different precautions you have to take, so that it will give you an idea about optimizing the protein purifications of your own choice, and as well as it may give you the idea how to perform the chromatography if you have the similar kind of samples. So, in this case I have taken the first examples where we are going to purify or we are going to learn how to purify the protein from the plant sources. So, the first example is that where we are planning to purify the proteins from the plant sources, and for this examples we have taken a plant which is called as Nigeria sativa. So, in the first step itself you have to perform the extraction. So, in the extraction procedures you have the different systematic scheme where you have to first make a watery extract of the plant, and then you dry and keep the powder, and then you can take the suitable amount of powder and dissolve it into a PBS at pH 6.4. Now, once you dissolve this into PBS, you are going to get, and when you vigorously mix this solution along with the PBS, you are going to get 3 fractions. In the fraction number 1, you are going to get the insoluble pellet, the pellet which is not going to be, which is going to settle down after you making the powder, dissolving the powder in the PBS. The second is there will be a oily layer which is present on the top, and then you have a soluble extract which is the fraction number 3. So, once you have dissolved this powder into PBS, you vigorously mix the powder, then carefully first remove the oily layer from the top with the help of the pipette, and then you spin this mixture, and that actually is going to give you the insoluble pellet as well as the soluble extract. This soluble extract can be dialyzed against the 50 millimolar phosphate buffer pH 6.4 and using a 3500 molecular cutoff dialysis bags. So, the dialysis membrane what you have to use in this case is going to be of 3500 Dalton molecular weight cutoff. Once you are done with this, you can estimate the protein, because you know that the every column has the a suitable binding capacity. So, you cannot load more than what is the binding capacity of a column. So, you have to estimate the protein using the Bradford method, and once you have know that you have the sufficient quantity of the protein, then you can take this and use it in the chromatography. So, the chromatography will be done with the help of the by packing packing the matrix into the column. So, in this case we are using the DI Cefadex A50. So, DI Cefadex, if you remember, it is a anion exchange chromatography matrix. So, once the protein is bound, then you can do the washing step, and after the washing you can elute this with a NaCl gradient in 50 millimolar phosphate PS 6.4, and whatever is coming out from the column can be detected by the spectrophotometric method by measuring the ODs at 280 nanometer, and that actually will give you the chromatogram to know in what fractions you have the proteins, and then subsequently what you can do is you can analyze these fraction in a SDS page, and that actually will give you the protein bands of the different molecular weight. Now, depending on what is your choice or what is your protein of interest, you can be able to refine and fine tune these protocol, and as well as you can be able to get the desirable proteins in a very high quantities. Now, in the next examples, we are going to purify the carbohydrates from the plant sources. So, for this what we want to do is we want to do the purification of the carbohydrate from the plants. For this we have taken an example of Olea europhea, and the steps are remain the same. You have to first do extraction procedures. For the extraction you have to follow a particular scheme. So, what you have to do is you have to first take the leaves as well as the root of this plant, and then you have to first prepare the water extract. The only advice or the caution is that the leaf or the root whatever you take should not be taken from the very old plant. So, it should be very up to the 2-year-old plant, and then what you do is you extract the watery extract with the D30 methyl glucoside and you extract it for the 15 minutes. After that you are going to get two fractions. You are going to get the insoluble pellet as well as the soluble extract. Now, the soluble extract you can just pass through a guard column, so that it should remove the impurities, because as you remember we have, while we were discussing about the preparation of the samples, it was very much advisable that you should not have the particulate matters, especially in this case where we have to use the guard column, so that you will remove all the contaminants, so that it should not interfere in the subsequent steps. After that you can do the chromatography. So, in this case we are using the not only one column, but the two columns. So, you can use the two anion exchange columns. One is called Dionix Carbapac P1, and the other one is called as the Carbapac M1 column. Both are the HPLC anion exchange columns, and the elution. So, what you have to do is first you load the soluble extract which you have passed through to the guard column first to this column, and then you do the elution with the help of the 12 millimolar NaOH containing 1 millimolar barium acetate. And once you got the fractions, those fractions can be loaded onto the next column, and that is how you can be able to fine tune the purifications. The detection can be done with the help of the pulse areometric detections, and that actually is going to detect the carbohydrates, and ultimately you are going to get all the carbohydrates whatever is present in this particular plant, and depending on the choice of your carbohydrate which you are interested to purify, you can be able to fine tune the purifications with the changing in the gradient curves, as well as the changing the pH of the solution, and as well all other parameters. and that is how you can be able to achieve the purification of the carbohydrate from the plants. Now, the third example is, because so far what we have discussed about the plants. Now, we will discuss about the animal cells. So, we will discuss how you can be able to isolate the protein from the animal source. For this example, we have taken the examples of the hen egg. So, our objective is to purify the protein from the animal source. We have taken an example of hen egg, and the first step as it is that we have to follow a extraction procedure. So, in the extraction procedure, what we have to do is we have to first collect the fresh egg, and then we have to break open the egg, we have to remove the yolk part. So, so first part is that you have to remove the yolk part, you have to just take the egg white, and then you have to do the isoelectric precipitation of the egg white with the help of the 100 millimolar NaCl. Once you do that, you are going to get two fractions. In the fraction 1, supernatant, which contains ovomyosin, and you are going to get the precipitate. This precipitate you can further dissolve into 500 millimolar NaCl with the stirring at 4 degree for overnight. So, once you dissolve this precipitate into 500 millimolar NaCl with stirring at 4 degree, it will again give you the two fractions. The fraction number 1 is the precipitate, which you are going to discard, and then you are going to get the supernatant. The supernatant, so the supernatant 2 as well as the supernatant 1 is going to pool together, because this will contain the protein, especially the ovomucin, and this one is going to contain all other proteins which are be part of the egg white, and then you pool these two. So, you pool the supernatant 1, you pool the supernatant 2, and then you use them into the chromatography. For the chromatography, in this case, because we have to fractionate the all the proteins present in the egg white, we have using the two column. One is called as the Q-Saffrose fast flow, which is the anion exchange chromatography column, and then you are using the sp-Saffrose fast flow, which is a cation exchange chromatography. So, in this, what you are going to do, first you are going to load the sample onto the Q-Saffrose fast flow, and then you are going to elute the sample with the isochromatic elution of the sample using the 0.14 molar NaCl. Following this, you are actually going to do the gradient elutions with the help of 0.14 to 0.5 molar of NaCl in the case of anion exchange chromatography. So, that actually is going to give you the different fractions containing proteins, and the unbound fractions. So, what will happen is, once you load the sample onto a Q-Saffrose fast flow and you will going to do the elution profile, some of the protein is going to bind, the some of the protein is not going to bind. The unbound fraction, whatever is going to you are going to get from the Q-Saffrose, is going to be loaded onto the cation exchange chromatography, which is the sp-Saffrose chromatography, and then you have to follow the similar elution schemes, which means you are going to first do the isochromatic elution of the sample using 0.14 molar NaCl, and then you have to do a gradient elution from the 0.14 to 0.5 molar, and ultimately you are going to use the mass spectrometry as a detector to detect the masses of the protein whatever is coming from the either the anion exchange column or the cation exchange columns, and ultimately you are going to detect the different proteins like ovoalbumin, ovotransferrin, lysozymes and ovomucin. And because all these proteins are of different molecular weight, so you will be able to detect them while they are coming from the elutions. So, these are the research problems where we are using or we are using the ion exchange chromatography to only purify the particular proteins. You can be able to have the multiple such examples, but overall what you have to do is you have to clarify the samples, you have to do an extraction procedure, so that your recovery of your protein from these samples should be as high as possible, and then ultimately you also have to perform the ion exchange chromatography, either the anion exchange chromatography or the cation exchange chromatography, or in some cases we have taken an example where you are using the combinations of the anion as well as the cation exchange chromatography to purify the many factors from the single source. Now, what we are going to discuss? We are going to discuss about the research problems where you can be able to utilize the ion exchange chromatography. So, this is the problem 1. The problem 1 is that the scientists have discovered a transcription factor in skin cells binds to the set of gene X, which is responsible for changing its color due to the exposure to sunlight. Now, what they want? They want to identify the transcription factor from the skin cells. So, what is the problem? Problem is that the scientists have speculated that there are transcription factors which are actually binding to set of genes or set of gene X. By doing so, it is actually changing the downstream expression profiling in such a way that it is actually changing the color of the skin. So, now before going into the solution of this problem, what you have to understand is what is the design of a particular gene and what is the function of a transcription factor. So, a particular coding gene in a mammalian cells is containing different components. For example, it contains a promoter, then it contains the exon or the introns. Together this region is called as the coding region. So, these are the only region which we are interested to discuss. Now, this is the region which actually gives the protein part, and whereas this is the region which is actually playing the role of the regulatory role, which means the expression of this particular protein is going to be regulated by the promoter, because the promoter is going to bind the transcription factors, and by doing the by binding of the transcription factor to the specific promoters which are bound or which are present to the next to the gene, it actually regulates the expression. But what are the transcription factors? The transcription factors are the proteins which are actually binding to the DNA, and that is how they are actually modulating the gene expressions. What you have to understand here is that they are not binding to the DNA, but they are only binding to the promoter regions. So, to solve this problem or to decipher the whole equations or to identify the transcription factor, what you have to do is, because DNA is negatively charged and the transcription factor has the positive charge region, because of that the transcription factor as well as the promoter is binding through the electrostatic interactions, and so you can be able to purify or exploit this phenomena, which means if you want to do this, you need a promoter, means promoter of this particular gene, and what you need is you need a source from where you are going to get the transcription factors. So, in this case the promoter is anyway belonging to the gene X, and the transcription factors you are going to get from the skin cells. Okay. So, what you have to do is you have to first break open the skin cells, you have to recover the mixture of transcription factors, and then you have to also clone the promoter of this particular gene, so that you are going to have the multiple copies of these promoters. Then what you are supposed to do? You have to design an experiment so that you will be able to control all the parameters, so that you will not be able to get the false positives. So, in this case what you are going to do is, so in a experimental design what we will do, we will take the DNA, we will bind it to the anion exchange column, because the DNA is negatively charged. So, it will bind to the anion exchange columns, and then what we will do is we will take up this transcription factor and allow them to glow, okay, and we will choose the pH in such a way so that the protein X what or the transcription factor what you are taking is going to be containing no charge, which means it will not bind to the column on its own. Then what you do is you wash this with the chromatographic wash with the salt, and then elute it with the salt. So, what will happen when you elute it with the salt, the DNA as well as the transcription factor is going to be eluted. Then what you do is you take this transcription, take the eluents and analyze it onto the SDS page as well as the agarose page. So, agarose is going to give you the pattern for the promoters, whereas this one is going to give you the pattern for the transcription factors. Now, the transcription factor, there will be multiple transcription factors which are going to be present in your lysate. So, if the transcription factor will bind to the DNA, or if a particular set of fractions are going to give you the binding, then the pattern of these proteins onto the SDS page is going to match with the pattern of the promoters. Okay. So, in this approach, the anion exchange column is incubated with the DNA and allowed it to the bind tightly. Now, the transcription factor is passed through to the DNA bound to the beads, followed by the washing with the buffer to remove the unbound the transcription factors. Now, the DNA is eluted from the matrix either by adding the high salt or with denaturating conditions. Either of these conditions. Now, what you are going to get? You are going to get the different fractions which you can test on the SDS page as well as the agarose, and that eventually will give you the pattern. That pattern is going to match if they are actually binding to the promoter region of that particular gene. As a control, you can also run the lysate onto the empty column without the DNA. So, whatever the transcription factor will bind to the beads and will come through into the elution profile, those are the transcription factor you can easily ignore, because it is not that the transcription factor is only bind, the other protein will also going to bind to the DNA. For example, the single stranded DNA binding proteins or helicases or DNA polymerase, these are the proteins also will have a very high affinity for your promoters or probably a small stretch of DNA. So, those proteins are also going to bind, but they will not going to show you a pattern, because their binding is going to be non-specific. They will bind to every region of the DNA, whereas the transcription factor is going to have a specific affinity, and they are only going to bind to the particular DNA. So, to execute these experiments, you need to perform 4 things. What you have to do? First, you have to clone the promoter of that gene X. Then you have to do the ion exchange chromatography, which means in this case you have to do an ion exchange chromatography. Then you have to do the agarose gel electrophoresis to see the pattern of the promoter DNA, and you also have to do the SDS page to see the pattern of the transcription factors. And ultimately, by doing this experiment, you will be able to identify a particular transcription factors. If you fine tune the purifications, you will be able to, or if you add some more purification tools. For example, if you use the gel filtration or affinity chromatography, you will be able to remove the outliers, and that is how you will be able to identify the particular transcription factor utilizing this approach. Now, let us move on to the next problem, and in the next problems, the problem 2 is that the cancer cells are treated with curcumin. So, curcumin is a drug, or curcumin is actually a phytochemical which is present in the turmeric. So, but curcumin is a very well known anti-cancer compound, and it activates the several protein kinases in the cell lysate. Now, a PhD student wants to measure the total kinase activity of the cell lysate, which means the objective is that the particular student wants to measure the kinase activity which is present in the cell lysate. Now, what you have? You have the cell lysate which contains the different type of protein kinases, and what these protein kinases are doing? They are actually converting the substrate to the with the help of the ATP to a substrate phosphorylated substrates, which means they are actually generating the negatively charged molecules or negatively charged substrates. So, in this case what you can do is you can easily perform the ion exchange chromatography to purify the negatively charged or phosphorylated substrate, and then you can be able to measure the kinase activity of the cell lysate. Now, what you have to do is, so what is the experimental design is that you take the substrate, okay, you radiolabel the substrate with the help of the carbon 14, okay. So, that actually is going to make this substrate detectable with the help of the radiography. Then you incubate it with the ATP. So, what will happen? The protein kinases which are present in the lysate is going to phosphorylate, and they are going to give you a negative charge, and in addition it is going to generate the ADP. Now, what you do is you take this complete assay mixture and load it onto the ion exchange column. So, in this case we are taking the ion exchange column like the cation exchange column. Okay. So, what will happen? The cation exchange column is going to bind everything except the negatively charged the substrate, which means you are going to, when you load the protein, when you load this experimental assay system, it will bind everything except the phosphorylated substrates, which means, and in addition it is also going to release the cold ATP and cold ADP, but cold ATP is hardly matters because what you are going to measure is the radioactivity associated with the carbon 14. So, carbon 14 which is non-phosphorylated is going to be trapped onto the column itself, whereas the phosphorylated carbon 14, a phosphorylated substrate, is going to be released from the column in the form of flow through, and then you can take up the flow through and measure the radioactivity. Ultimately, you can also do a elution with the salt, and you can be able to measure the radioactivity to confirm that the column has trapped the radioactivity. As a control, you can also do a control reaction where you can do everything, but you can remove the kinase from the reaction. So, if you do a removal of kinase, whatever you are going to get the counts, these are going to be the basal level counts, and these counts are going to be subtracted from your experimental counts. So, for this particular experiment, to perform this experiment, you have to prepare the cell lysate, then you have to incubate the cell lysate with the radio labeled carbon 14 substrate, and then you also have to add it to the ATP and incubate it for some time, and then you have to perform the ion exchange chromatography. After this ion exchange chromatography, whatever the flow through you are going to get, that flow through can be analyzed in two different ways to get more and more different informations. So, what you can do is this, you can analyze in two different way. One, you can easily collect the you can take up the fractions and you can do the flow through and you can collect the radioactive counts, and that, so you can collect the radioactive count in the scintillation counter, and that actually is going to give you the kinase activity associated with the cell lysate. On the other hand, you can also analyze these flow through into a SDS page, and SDS page followed by the identification of the substrates. So, if you do the SDS page and then you further you do the autoradiogram, or if you do the Western blotting, you can be able to even identify the substrates. So, we started with a single experiment, but that single experiment can be diverged in a multiple experiment, and that is how you can be able to get two different informations, like what is the kinase activity associated with the cell lysate and what are the different substrates which are present or which are going to be get phosphorylated by the these kinases. So, these are this by diverging the one single reactions, you can be able to get more and precise information about the experimental conditions. So, with this we would like to conclude our discussion about the ion exchange chromatography. What we have discussed about the basic principle of ion exchange chromatography. We have also discussed about the different steps what you have to do and what are the crucial parameters you have to consider when you would like to utilize them for running the ion exchange chromatography, and also how the different parameters are crucial to choose the specific matrix as well as the specific pH at which you have to perform the ion exchange chromatography, and what are the different buffers which are available for cation exchange chromatography as well as the anion exchange chromatography. And in today's lecture we have also discussed about the potential of the ion exchange chromatography in the solving different types of problems, and how you can be able to use the ion exchange chromatography to purify the protein from the complex mixture. So, with this I would like to conclude our lecture here. Thank you.